What is going on? It is me, and it is February 3rd, 2022. You might have known if you were paying attention to my channel that I didn't, or noticed that I didn't post a video yesterday, and that's because while I was ready to post a video, I did not feel comfortable with my knowledge absorption for the day regarding the brain cells and the support cells of the brain besides neurons. And that's what I was going to talk about yesterday, like the glial cells, the oligodendrocytes, the Schwann cells, a few other support cells like microglia, some pericytes, and astrocytes, and the myelin itself that is generated by the Schwann cells, which in turn come from the oligodendrocytes. I was going to talk about all that yesterday, but I realized that I really need to dig into those pieces a little deeper because there's quite a lot of architectural organization going on in the brain. And many organs have some complex, anatomical organization, and especially when you get into the deeper level of the actual biochemistry that goes on within the actual cells. But the brain's like upper level organizations, the neurophysiology is unsurprisingly quite complex, right? And today I wanted to actually take a step back and talk about the blood-brain barrier a little and astrocytes. The reason why is because it's better to work our way into the brain because the brain is quite complex in terms of, it's funny because when you think of the brain and in school, they kind of really don't go into it a ton. You think of only neurons, right? But or in like high school or whatever. But the neurons themselves, they have a relatively individually simplistic function, but there's so much that goes on between different neurons and the glial cells themselves, which normally get ignored, that you kind of have to understand how everything fits together and repeat the process of understanding that. At least that's been my experience so far. So with that being said, I will start with the astrocytes and blood-brain barrier because I did want to talk about the glial cells. But let's talk about the BBB. So the blood-brain barrier is basically what it sounds like. It's a barrier of endothelial separation between the space, the extracellular space within the brain, and the blood of the rest of your body, your bloodstream itself. And the blood-brain barrier, again, it's if you look at it, it's an endothelial layer that surrounds the blood vessels that surround the brain and essentially it prevents certain substances from getting into the brain. So the brain really needs, obviously, blood, right? Like every organ needs blood, but the brain needs oxygen. It needs all your standard chemicals of life and chemicals of source chemicals to survive, which include oxygen, carbon dioxide, glucose, certain proteins, certain ions, and whatnot. But the blood-brain barrier really is designed in a way to only selectively let things in, because when stuff gets into your bloodstream, there's quite a lot of stuff floating in your bloodstream. If we ever go into breaking down every component and everything that could possibly be in your blood, including pathogens, you'll see that that a lot of things, if there was no selectively permeable barrier preventing excursion into your space or incursion into your brain space, then you couldn't survive. It would be absolute chaos, because your brain completely functions off electrical impulses, right? All of your neurons are sending electrical signals everywhere, and if you know anything about electricity, you should know that electrically driven systems are very sensitive, especially on the microscopic scale, right? Like if your brain requires a certain charge distribution to make a decision, right? Like ultra simplifying it, but it makes sense, I think, for this example. If a little protein comes by and starts like slightly altering that charge externally that was sophisticatedly calculated by the rest of the neurons in your brain, then the end result is not going to be what was intended, and your thought would be different. Your neurophysiology result would be different. Whether your muscles get stimulated properly would be different. Everything would be different because your control center would be uncontrolled itself. So essentially, the blood-brain barrier really has to be very, very strict in what it allows into your brain. And how does that do that? So your endothelial cells are essentially there's just standard endothelial cells with they have your phospholipid bilayer there, right? And whatever's in your blood that's not lipid soluble, right? Small and lipid soluble, two criteria, will not be able to pass through into the blood-brain barrier. So most of the lipid soluble substances that can pass through small lipid soluble substances are, they fall in the category of oxygen, carbon dioxide, things like that. And those are important because those are nutrient and gas exchange, waste excretion, right? Your brain excretes carbon dioxide into your blood. Your brain uptakes oxygen from your blood, and that's just some basic components needed to survive. It's needed for you to survive. And needed for your neurons to survive. And what else is needed? Well, there's ions that are needed to. Ions need to traverse your brain, right? And also other sugar, glucose needs to traverse your brain. So the blood-brain barrier actually has specific channels that allow transport of non-lipid soluble molecules through the BBB. So, like for example, if you need to transport a protein, or if you need to transport glucose, you have glucose transporters within the BBB, which allow the blood-brain barrier, to open itself up to the glucose to enter selectively into your brain space and be transported to where it needs to be to carry out cellular respiration, right? Because the Krebs cycle depends on that. So this is another view of kind of like how the blood-brain barrier is kind of like surrounds the blood vessels, because imagine these are blood vessels going into your brain, and you have the smooth muscle, which is like essentially the endothelium. So if you take a cross section of that, let's take a cross section of the endothelium, right? So the endothelium is this part here, this endothelial cell, this innermost layer, right? This innermost layer, actually the endothelial cells are, and this is very important because how does your brain, your endothelial cells allow, like this, your endothelial cells are of a certain size, but after that, right, they have to connect to each other in a way that prevents leakage or sneaking of molecules past them. So what happens is the endothelial cells are connected to each other by something called tight junctions, right? And if you look at tight junctions are they're these things that are in between the endothelial cells, right? And they have a ton of different factors that go into stimulating their creation and stimulating their dissolution. It's quite complicated, as most things are in biochemistry. But either way, the tight junction formation is at a higher level stimulated by your astrocytes, right? So your astrocytes essentially connect to your blood vessels, and your astrocytes have many different important functions. At one point people didn't think astrocytes did anything but support the structure of your brain, but astrocytes are important for nutrient exchange, and that's why these branching elements of the astrocytes, these dendritic elements or foot processes as they're called, connect to your blood vessels through the blood-brain barrier, right? So, and what happens is these astrocytes, the foot processes on the astrocytes, tell the blood-brain barrier to create more tight junctions when the permeability of the BBB is to be decreased. Okay, so these, and just kind of giving you an overview of what's on top of what, right? These epithelial cells lie on top of what's called basal lamina, right? And the astrocytic foot processes actually touch the basal lamina. Basal lamina is just connective tissue. Okay, it's nothing like crazy, and you have like lots of laminar connective tissue in your body, but that's basically what happens there. So like, yeah, if you go down to another visualization of what this looks like, this is like your cell has your basal membranes, which have basal laminar tissue, and your skin cells rather, and these are the epithelial cells. Another section, another more zoomed in version of the basal lamina, right? And the astrocytes, you can imagine, have their foot processes connected to the top layer of the basal lamina. Like in this case, this would be the lamina densa, etc. That's not super important for what I wanted to talk about, but here's the other thing. Let's go back to so you can imagine these are the astrocyte end feet that stimulate tight junctions, so that stimulate the connection of this endothelial cells to other endothelial cells, right? So you see this blue part here. This is something that's not talked about that often, right? But it is important because it's needed, but it's not like, it's sort of in between the vascular system and the neural system, your neurological system. Some, I believe, refer to it as the neurovascular component, or part of your neurovascular system. What happens is this is called a parasite, and this parasitic cell, this parasitic cell helps with constricting the endothelial cell and the blood vessel selectively, right? So there is, that the parasites surround the basal lamina. And if you look at, there was a good image somewhere around here on how the parasitic action, here's this is kind of like these are astrocytes you see astrocytes, how populous they are, right, in terms of their foot processes, how, like, where they connect to. But the parasite essentially kind of surrounds the endothelial cell, and they wrap around and they kind of help blood, with respect to the brain's stimulus, flow in certain ways. So again, there are more support cells, right? We'll kind of talk about those later. We don't need to talk about them right now, I guess. And this is another example of where the parasite might live. But let's talk about what else we need to know here. So the astrocyte also, so that was the blood-brain barrier in a nutshell. There's actually quite more to talk about there, in that there's many diseases. Like I think I mentioned multiple sclerosis as a disease that circumvents. It's an autoimmune disease that essentially it is the result of your blood-brain barrier being circumvented by your T cells, and your B cells and other lymphocytes that somehow sneak into your brain space. Okay, so that's basically it. And then at some point, maybe tomorrow, we'll go into astrocytes. This is interesting. This is kind of like an interaction between a reactive astrocyte and your cytotoxins killer cells, right? So this is your astrocyte kind of fighting with your own cells, right? Your own police that shouldn't be inside the brain space, right? So that's why this is an example of like an autoimmune interaction, right? Kind of interesting, this kind of thing that's happening there. Okay, and we will talk about the other functions of the astrocytes, because this is the blood-brain barrier that we talked about, lipid-soluble molecules that need to pass through, and selective permeability due to astrocytic stimulation of endothelial tight junctions is the other kind of like interaction which allows permeability to increase or decrease. between endothelial cells, and then the transport channels within the blood-brain barrier, which essentially are needed to be considered when designing drugs for the brain, because if you want to build out something that can penetrate the blood-brain barrier, you're going to actually have to use one of the transporter channels to do that typically. And that means that you're going to have to either have your drug packaged with a protein that will trigger the blood-brain barrier channels, the transmembrane channels of the endothelial cells of the blood-brain barrier to allow your drug to enter, or you're going to have to do some other tricks that drug designers do. We might get into those at some point. But yeah, and jumping back to my final point for today, I just wanted to talk about the astrocyte, other functions of the astrocyte in brief, which I'll get to later, which revolve around neurotransmitter regulation. So, and axonal action potential regulation as well. So essentially the astrocyte will clean up and recycle excess neurotransmitters. Okay, it will also clean up and recycle excess potassium that leaks out during an action potential propagation, because remember, during propagation of your action potential, potassium is exchanged with sodium, right? So that there's a process of a transference of charge across the axon. But sometimes what happens is excess potassium exits from the cell, and you have something called ATPase and Na plus potassium, sodium potassium ATPase, which helps during the rectification phase, which is after the depolarization phase of the of the action potential of the neuron, to allow some of that potassium back in. The potassium doesn't always go back into it doesn't go back to where it should be inside the axon. So that excess potassium is actually sucked up by the astrocyte and stored for later use, right? Like the excess potassium may be needed during a potassium downturn, like if you didn't have enough bananas or something. And then the astrocyte can then supplant that extra potassium need for thinking that has to happen. So, and then again, the neurotransmitter element, the neurotransmitters, remember, they have to be recycled after they're used. For example, we went into the neurotransmitter acetylcholine process, and acetylcholine is excreted from the synapse into the synaptic space of neurons through packaged vesicles which dissolve with the synapse. That excreted acetylcholine neurotransmitter within the synaptic space connects to the muscle cell, and remember there's receptors on the muscle cell which let ions in, sodium in. Those, as long as that acetylcholine is trapped onto those acetylcholine receptors, the action potential will keep increasing and continuous stimulation will happen. So something called acetylcholine esterase has to come by and break the acetylcholine into an acetate and choline, which the choline then gets transported back into the synapse. This happens for not only acetylcholine but also the other neurotransmitters like glutamate and GABA. Glutamate is a specific example of something that an astrocyte will, through several chain processes, take in and store, and it will. And GABA is also created from glutamate, so the astrocyte recycles for gluterogenic neurons, ones that use glutamate as a neurotransmitter. The glutamate is recycled through several steps back for those gluterogenic neurons, and then for the GABAergic neurons, which use GABA, which is a neurotransmitter as well. The glutamate, it synthesizes the glutamate needed to glutamine needed to create GABA itself, and so those are GABAergic neurons. So that glutamate is kind of used in 2 ways or 1.5 ways in neuron and neurotransmitter recycling. So yeah, that was a mouthful, and I got a lot off my system, but it was a longer off my chest, but it was a more than I had intended, and it made up for yesterday, I believe. So with that being said, I want to bid you adieu. Have a nice day and make your neurons happy.